Okay, so Lee mentioned that he felt a little bit out of water, so to speak, with this session, and I'm actually even more out of water to a certain extent. I'm an evolutionary biologist who's focused on aquatic systems through my course of my career, and I want to talk a little bit about a recent project that is ongoing that's been funded by the National Science Foundation, and we're using this approach called resurrection ecology. And my study organisms are freshwater zooplankton, in particular water fleas in the genus Daphnia. So we've heard talks so far today about how global environmental challenges are out there. There's change going on. It's more than just climate change, of course. And the one area that we've focused on quite a bit is the role of fertilizers and nutrient pollution to aquatic systems. And as shown in an earlier slide that Lee mentioned, we can get these cyanobacteria blooms. This is a photo of a lake in western Minnesota. It's part of the Minnesota Department of Natural Resources Sentinel Lake program. And here's a bloom of Aphanizomenon. And what we did was, over the course of the last 9 or so years, we've been looking at using this approach of resurrection ecology. What this means is lots of organisms out there, microbes, plants, organisms like my Daphnia here, the water flea, they produce resting stages that can go through long periods of diapause. And so they rest in wherever the egg bank, the seed bank, or whatever, and you can resuscitate these organisms, basically do time traveling. Return, you can go back in, hatch them out, germinate them, and return organisms that have not been around for a very long time back to modern times. And so what we've been doing is we've been hatching out these resting stages of Daphnia. And here's an example that for some of you may know Daphnia from the ecotoxicology literature. It's been an important bioassay organism for over 100 years. And what we like about using this organism, it's got some features that are really pretty neat. First of all, it's got very short generation time, 7 to 10 days on average. It's small. In fact, some of my colleagues in the fish realm say it's not really an animal, it's a light diffraction. I tend to argue with them. Easily cultured. It's neat because it's got a mixed breeding mode, clonal reproduction, so it basically clones itself, and it can also sexually reproduce diapausing eggs. There's genomes that are out there, so we're also looking at genomics and related to how organisms like Daphnia can handle environmental stressors, like phosphorus, for example. And then, as I mentioned, it's used in ecotoxicology extensively. All right, so the study sites, we focused on 5 lakes in particular in Minnesota. I'm only going to focus on South Center Lake. This is our flagship lake. And what we do with resurrection ecology, we go, before I do that, I'll show you a little bit of the relationship between the watershed in South Center Lake and the area around it. So this is a plot from a paper that we published a few years ago. This shows the historical agricultural use in the watershed in Chisago County, Minnesota, around Center Lake. Here you can also see the plot of phosphorus buildup in the sediments as a major nutrient pollutant. This shows here a very interesting pattern. This was the late 1970s with the oil embargo that occurred. Acreage dropped, and at the same time you see a drop in the phosphorus signal in the lake. It rebounded a few years later. So using these proxies to look at changes is what we do. So we go out to a lake, we take sediment cores, we use these nested sieve series here to fish out these eggs, pop them open, incubate them, to hatch them, to raise these cultures of Daphnia. This is a very complicated slide, but I'll go through it really quickly. What we've been able to do is go through time. This is the surface down, whoops, hold on. Surface all the way down here through the sediment core. We see genetic changes here. The greens show phosphorus changes, and these areas here show Daphnia that we've been able to resurrect. The oldest Daphnia we've been able to resurrect are centuries old, predating any colonization of the area and any use of fertilizer and so forth. We can then interrogate these Daphnia, looking at physiological changes through time. So these are modern and these are ancient lineages, looking at how they retain phosphorus and other additional important environmental cues. So my point is that using resurrection ecology may help us understand how organisms are evolving and adapting to environmental change. And I want to thank you and bye-bye.